Okay, thank you.
Yeah, I think it's done now. Yeah, you can go ahead, sir.
Yeah, thank you, Dr. Ravindra. Shakil, you can start.
Yeah. Good morning, everyone. Thank you for joining us and thank you for your patience. We had some technical glitch, so that is why we're starting a little late. Today is week second for...
Shalvia?
Yeah.
Do you want to wait for some time because there is no one on the stream right now? Because it got activated just now, so it might take some time for people to join in.
Right, but I think they can start from the beginning or no?
They can, but the Q&A would be delayed by...
Oh, okay, so
There are some 226 people, I think, 1 minute. There are some good number of people I can see.
It's okay. We are on time, so we can start. Not a problem.
Yeah, it is better to start. Like otherwise.
Sure, go ahead then.
Okay. Thank you for joining us. Today is week 2 for Indian webinar series, Introduction to Computational Drug Design. It is co-organized by Schrodinger-PCI. I would like to thank our collaborators for supporting us and also all the panelists and the IT team of JSS University for hosting this live session, and the IT team of Manipal for hosting these sessions on their channel as well. Today we have 2 sessions. The first one is when the target protein structure is not there, what to do in that case, and molecular dynamics, simulation theory and analysis. So I think without further ado, we'll start with the first session. During this course of presentation, if you have any questions, feel free to drop your queries in the chat section of the YouTube link. Your Q&A session, the questions will be addressed during the Q&A session after the presentations.
Dr. Ravindra, the live streaming is not appearing. Can you please confirm once? In the university YouTube, I don't see any live streaming going on.
Just a minute. There appears to be some issue.
Did it start at all or did it stop in the middle?
It is going on JSS channel.
Okay.
So I don't know about others.
It doesn't display on the homepage of JSS. The stream is active though.
Yeah, even I can't see it on their channel.
If you have the Zoom session, then you can see it by clicking on live on YouTube, but I don't think you can access it otherwise.
Okay.
He has left.
In the meantime, can someone make me a co-host? So get done with that.
I'm not able to do that. I tried that. I think host only has to do it.
Kaushik, don't worry. You can present
Another
this.
2 minutes. I think, somebody else is streaming on the same link. That's why, it's not permitting me. I'll just get,
Okay.
get that right. Give me a minute.
And we are starting to get emails for the link.
Yeah.
I think we are there now. Somebody else had taken over that. Now I had to, get them off.
Okay.
Can you just verify once? I think one of you can
Confirm, yeah.
confirm.
No, Ravindra, it is still not there. I don't.
No, it
It
is
came,
there.
sir. Your YouTube link is came. No problem.
Sir, it
There,
is
sir.
coming now. It's coming. Hello?
Just checking, sir. 1 second.
Yeah, I think now I can see.
Still not, sir.
I don't see it.
Yeah, I too don't see it.
Sir, I'm able to see it. There are some 20 odd people are already there.
See it now on the homepage.
Yeah, now it is there.
It is there, no?
Yes.
Okay. Please carry on. Sorry, my apologies. I think there's some issue.
No problem. Happens. Shall we? You should restart from the beginning.
Okay. Good morning, everyone. Thank you for joining us today. Today is the week 2 for Indian webinar series, Introduction to Computational Drug Design. It is co-organized by Schrodinger and PCI. I would like to thank our distinguished collaborators for supporting us, and also the IT team of JSS and Manipal for hosting these sessions. So without any delay, I think we should just start with our first session. When the target protein structure is not there. During this course of presentation, if you have any questions, please feel free to drop them in the chat section of the YouTube link. We will have a Q&A session after the presentation.
Shall I start, Shelvia?
Yes, Dr. Prashad, please start.
Okay,
You're very
great.
good.
Okay, thank you very much. So let me share the screen first. Yeah. And here is the full screen mode. And let's see. Yeah, can anybody confirm like it is
Yes,
visible
visible,
and audible?
audible.
Okay, great. So thank you very much, Shelvia. Uh, and thank you very much all of you for joining the session. So as today we are discussing about the problem when you don't have structure of protein available with you. So what to do in such case? So in drug discovery, like there can be many various cases and conditions which might obstruct you to perform further research. But again, it is not really the thing. uh, you will have all tools available with you to work with to resolve your issue. So one of such tool is homology modeling. So what is the thing here? Whenever you don't have protein structure available with you, so in that case you can perform homology modeling for your protein structure and you can proceed further for your drug designing project. So this is what today we are going to discuss about the homology modeling and its application. So I'll start from really something which is basics and we'll proceed through how to perform homology modeling at the end, and we will have separate discussion session on it, whatever questions you will have. So stay tuned. So this is what is my classical slide of basics of drug designing. So as I always mention, it follows the lock and key model, and the drug discovery research is all about finding the best key amongst all keys available in the database. But to perform all those exercise, you must know the structure of lock. So there can be condition when the structure of lock is not available. So what to do in such case? You need to generate a structure of lock. So same thing is applicable in case of protein or your receptor. So whenever you don't have structure of it, we need to develop that structure so that we proceed for further drug designing studies. So as there are various receptor based or ligand based methods available for you to perform your drug designing research. So we have seen many of such examples of structure based drug designing, and in consecutive lectures we will see some ligand based drug designing or higher level of molecular modeling studies like molecular dynamic simulation studies. So but here in this presentation I'll be mostly focusing on homology modeling of the protein structure, and once you will have homology model of protein structure ready, you can proceed for the structure based drug designing. So this is what the idea about. So what you can do with protein modeling. So these are the tools which are available with Schrodinger suite. You will have all those tools available with you, and you can basically build the structure of your target protein using homology modeling. Then you can analyze that particular structure using site binding site identification or sitemap tool, which we have seen before. You can analyze how it looks like, how the protein structure look like, how binding site look like, and you can use that information for further molecular modeling studies or computer aided drug design studies. You can also refine your protein structure, or not only it is not only about the refinement of your homology model, but you can also refine whatever crystal structure is available with you. So that tool is also available in the Schrodinger. For example, if you have a crystal structure of low resolution and you want to refine it further, that also you can refine. And you can also perform molecular dynamic simulation studies on your protein structure or biological target or protein ligand complex to refine it further or to study it further. So these are the various things which are available with you with the Schrodinger suite. So now let's start something about the basics. So basics of protein means how protein is made up of. So basically protein is made up of 20 amino acid residues. So there are many such residues, for example alanine, cysteine, aspartic acid, glutamic acid, and many. So this number is, there are 20 naturally occurring amino acid residues through which protein is made up of. So whenever two amino acid residues get together, they form a peptide linkage, which we call it as CONH. So peptide linkage joins two amino acid residues, and in this way this polymer continues to grow, and once it grows completely, you will have a tertiary structure of protein. So it ultimately gives you the structure of protein. So at very basics, you need to understand how it is made up of, what are the building blocks of your protein structure. Means if you start it from very basics, you will able to understand how it is and how you can use that information for further your drug designing studies. So let's see, there are few terms which we need to understand in case of protein. So basically, whenever you have a sequence of amino acid residues, for example, all that amino acid residues which are abbreviated in single letter code. So whenever such sequence is available, we called it as a primary sequence. So you can say it as a FASTA sequence also. And whenever this all amino acid residue join together through peptide linkage, which is CONH, it forms a long chain. So that long chain has no structure available, no structure present in it. It is just a simple polymer chain. So we call it as a primary structure of protein. But as it started growing, as it environment favors the folding of this structure, it folds into various substructures, which we called it as a secondary structures, and those secondary structures are alpha helix or beta sheet structure. So it looks like it has a helical turns, or beta sheets are some parallel strands which comes together. So it forms secondary structures. So whenever we say secondary structure, means some definite structure is available with us, and there can be various loops which are kind of distorted, and there is no structure present with loop. It just folds like a thread, and we call it as loop. When all those things come together, like alpha helix, beta sheets, and loops, all together, they form a tertiary structure of protein. So from primary structure, it ends up to secondary structure further folds to the tertiary structure, and this is the functional form of protein which you will have from, let's say, protein data bank or from homology modeling, is the tertiary structure of protein. It has definite shape, it has definite volume, it has definite function to perform. So this is what is our biological target. But even sometimes those all tertiary structures, like protein structures, come together and they form a quaternary complex. Means there can be number of tertiary structures come together and form a big assembly. That assembly also has specific function, specific structure. We call it as a quaternary structure. So simple example is a virus capsid. So virus capsid is made up of lot of proteins which come together and form a quaternary structure. So, or actin filament, or there are many such proteins which exist in the quaternary form, because it needs to perform some function. So that's why they come together and form bigger structure. So this is what is the basics of how the structure grows. And there are many such experimental methods through which one can find out the structure of protein. So those methods are very popular. Method is X-ray crystallography, through which experimentalists map the electron density and find out the structure of protein. So this is one of the method which is reported in protein data bank. So you will find many of such structures which are solved through X-ray crystallography. Then there is another method which is NMR method for structure resolution. So basically NMR is because it works only in the solution form. So you will have the protein structure in solution form, which is highly flexible in nature. So there can be various conformations of loops present in the same structure. In case of wherever NMR structure is available. NMR structure also has some limitations, like whenever protein is less than 30 kilodalton, then only it works. If it is more than that, then one need to go for X-ray crystallography or other methods. So There is also one of a good method is cryo-EM method. So through cryo-EM, actually one resolves the big assembly of structures like quaternary complexes. So those structures are also being solved through cryo-EM method, and nowadays this is very much popular method for solving big protein structures. So whenever you visit protein data bank, you will find various experimental methods through which structure has been resolved. So you will mostly find the X-ray, NMR and cryo-EM methods, and whatever is available with you, one need to decide which method, which structure I should go with. But overall X-ray crystallography gives you good kind of resolution that you can use it for further drug designing. So which are those databases where you will find all those structures? So one of the very popular database is protein data bank. So you can see PDB. So where you will find mostly all of those structures, but it is not only limited to protein data bank. There are many such databases like protein data bank Japan or European protein data bank. So there also you can find protein structures where experimentalist normally deposits their structure once it is published. So you can always look into such databases where you can find the experimental structure to work with. So what if this is what the idea of this today's session is, what if you don't have the structure of protein available in the database? So normally if you find it in database, that's well and good. If you don't find it in database, then you need to ask question to yourself, can I develop a model for this particular protein? So the best answer is you need to look into the sequence database. Sequence database is kind of UniProt database. If you have a FASTA sequence of your protein available in UniProt database, then yes, the answer will be yes, you can develop a protein structure using homology modeling method for the protein of your interest. So first thing you need to go to is UniProt database, and there you need to search for the name of protein of your interest, and you need to find whether the amino acid residue sequence is available with you or not. So in the basics of homology modeling, like once you have a structure of once you have a sequence of amino acid residue sequence of your protein, then you can go for template based homology modeling. So the very basics of homology modeling is just one statement, like whatever is, if sequence is similar, then structure is also similar. So you just need to remember this term, sequence similarity is equal to structural similarity. So similarity means whenever similarity comes into picture, it always refers to the two different things, means you need to compare these two things. And what are those two things is the template structure and the target structure. So this target structure means your unknown structure for which you don't have a structure available in any databases, and you wanted to build the model for this particular structure. We call it as a target structure, which is unknown. And template structure is the structure which is available, which is already available in the protein data bank, but it is somehow similar to that of your target structure. So you need to basically find out that best template structure available in the structural database, and then, as I said, if sequences are similar, structure is also similar. So if you see like sequence is similar in your template structure and your target structure, target sequence, then of course you can build the homology model of it. So you just need to align those sequences to see how similar it is. If it is having good similarity, then yes, you can build the model of your unknown protein structure. So that's what we call it as a homology model of the protein sequence. And here you can see whenever we generate a homology model, it almost generate the similar type of model whatever is available in the template structure. So here you can see orange color and green color. So green color is basically your template structure which is already available, and you try to build the exact similar structure as that of template structure for your target structure, means for which you wanted to build a homology model out of it. And in this way it just build the structure as just similar as that of template structure. So there are various steps which you need to follow for doing the homology modeling, specifically template based homology modeling. So what are those things? What are the steps you need to follow overall? So you can see it's a simple protocol which I have outlined for doing homology modeling, which starts from, as I said, first thing you need to ask is whether protein sequence is available in the database. So you just need to go to UniProt database and search the protein of your interest and see if you have sequence available, amino acid residue sequence available in for this particular protein. If it is available, then yes, you need to retrieve that sequence. Once you will have that sequence with you, there is a method called BLAST-P, means you need to find out the suitable template structure for your homology modeling. So for that we run BLAST-P search. This is nothing but the search algorithm to find out the best suitable template structure out of it. And at the end of this search things, you will have various parameters to consider. For example, how much is the sequence identity and similarity between template and target, how much is the query coverage, how whether there are missing residues present in template, what is the resolution, like if it is X-ray crystal, then what is resolution of this particular template structure, whether it consists of ligand present in it or not, or what is the PDB ID of it. So all those things you need to consider before choosing a final template structure which comes out of this BLAST-P search. So once you will have the best template available with you, you just need to align those two sequences using the software. And whenever you align those sequences, there can be various methods, like you want to go for single template alignment or multiple template alignment or secondary structure based alignment. So these are the things which you need to consider while doing target template sequence alignment. So once you will have best alignment available with you, you just need to go for model building. That's the last step of model building. So even for model building, you will have different methods like knowledge based method or energy based method. Or do you want to consider ligands? Means do you want to import whatever ligands present in template structure into the target protein structure? For example, if there are cofactors like NADP or ATP, so such things you can import inside the homology model. So you can also further build your backbone or side chain amino acid side chains of the amino acid residue. You can add missing loops or side chains in it. So or you can generate various chimeras. So these are the things which you can do using homology actual homology modeling exercise. So once you will have a model available with you, you need to check that model whether it is correct or not, whether it is reliable or not. So for checking of the protein structure, there are various things available. So for example, protein reports which find out which are the whether there are any steric clashes or whether there are any deviations from the Ramachandran plot, which is one of the classical method to analyze protein structure, whether it is correct, good or bad. So then there can be, you can compute RMSD, you can analyze the binding site for validation of your homology model, whether it is good or bad. So these are the things. If it is good enough, then yes, you can proceed further with this particular model. But let's say if it is not good enough, if it has many steric clashes or deviation to Ramachandran plot, then you need to definitely refine that model. So there are various methods to refine that model. For example, there is loop refinement or energy minimization or rotamer exploration. So these are the various methods through which one can refine the model. Once you refine your model, again you need to go back and check whether it is correct or not, whether it follows all those criteria or not. And if it satisfies all those criteria, then yes, you can proceed further for the for your drug designing studies using that homology model. So this is what the general protocol which you need to follow in building your template based homology model. But again, as it looks like very complex, but these are the all things which works in a background, and you will have simple tool to build a homology model in Schrodinger soup. So it just 3 clicks panel in which you just need to 3 times you need to click on button, and then that's it. It will generate a homology model. for you. But these are the various things which you need to consider, and of course software considers all those things in the background. So at some point you really don't need to worry about it. Software takes care of most of the things. But remember, these are the various things which are working in the background. So basically how this particular homology modeling is being performed. So as I said, like it is because it is template based. So whatever, if sequence is similar means structure is similar. So what software does in background is it aligns your target sequence to the template sequence, and once it aligns, it looks for the how much query coverage is, which amino acid residues are being aligned to each other, how many similarities is there, how many identical amino acid residues are there, and whatever is present in the template structure, it tries to copy all those information about amino acid residue sequence in your protein structure, and somehow it builds your target structure based on the template structure. And here you can see in template structure, if you find helix, in target structure also it will build helix, and in this way if it finds a beta sheet, then in target structure also it will generate a beta sheet. Again, the complex algorithm works behind it, and finally it gives you the similar type of structure as that of template structure. And you get, once you get that model, you need to go for further refinement or validation of this model. And again, this is what is the iterative process which one need to follow to build a exact, good, reliable homology model. So there are some things which you need to consider while building homology model, that it requires a good template structure. So because everything is being copied, or everything is being learned from the template structure, so it should have good template structure. That you can get it from the sequence alignment or blast search, and those various parameters which we discussed before. You need to consider all those parameters to find out the best template structure out of it. And once you get the template structure, you need to align it properly, because this alignment is very, very crucial. Like based on alignment only, it is going to copy all those things which are present in the template structure into the target structure. So that's why alignment is very, very crucial, and one need to take a look at alignment, whether it is good or bad. Then very important thing come into picture is the force field. That also you need to consider. You should be dealing with the best force field out of it, to which, because force field is the one which governs all type of interactions, bonding parameters, non-bonding parameters, which interactions are being happened inside the protein structure. All those things are considered through force field only. So that's the reason why you need to consider all those things. So as I said, like it requires a good template structure. So template can be, first of all, it can be a single template which is available for your structure. If it is single template, that's very well and good. If you are not happy with single template, you can also go with the multiple template structure, or you can generate a consensus model out of many various template structure for generating a single model. You can also generate chimeric or composite model out of your protein. For example, there is a particular region which is differing in the protein structure. For example, here this is the protein structure, and this green color domain is only being changed, and rest of the things are being same. So in that case also you can generate a chimeric model out of the homology modeling method. You can also generate a dimer or multimers in case of big assemblies or quaternary structures. There is a method for GPCR specific modeling, because GPCRs are the structures which are transmembrane structure, and it needs a specific treatment. So specific treatments need to be incorporated in the software, and with Schrodinger you have that option available for GPCR specific homology modeling, and most of the software lacks such option. Then there is a loop modeling which comes under refinement of the structure, and you need to perform loop modeling in very nice way to get a reliable model, because loops are the distorted structure and one need to take care of it. It is very, very critical. But again, as I said, with Schrodinger everything comes handy. Like for each option you will have a single tool which is single click button thing, and it will take care of everything. You just need to decide which model you need to go further with. The next thing comes into the sequence alignment. As I said, it is very crucial. So you can see here the sequence alignment of various kinase proteins, and once you align this many sequence, you will find which amino acid residues are differ between two sequence. For example, here in the at this particular region there is difference, though this particular valine residue is conserved amongst all proteins, but still in two proteins it differs. So you need to, based on that information, you need to think which template structure I should go further with, or there can be various regions which are missing in your template structure, so that also you need to think of. There are various anchor residues. For example, in kinase you will have anchor residues like DFG loop, which is present in every protein structure. As you see, it is well conserved, so it should align to each other. So this is what alignment means. You need to have that perfect alignment while building your homology model out of it. So not only that, but you can also have information about which ligand is present inside this particular template structure and what is the biological activity of it. So if you look into biological activity, you should go with, if you are going for further drug designing studies, then it is worth to go with highest active ligand molecule which is present inside it, because it represents the bioactive conformation of protein and ligand. So that's why the activity, biological activity of ligand is also critical in such cases. So once you will have that good template structure and good sequence available with you, so next critical thing comes with the force field, because as I said, force field is the thing which governs all type of bonded, non-bonded interactions. Whenever two atoms are bound to each other, it is a harmonic bond, and if three atoms are there, then it's an angle which is there between three atoms. If there are four atoms available, then it's a torsion angle. Or even the atoms are not connected to each other, it follows the various non-covalent interactions like van der Waal or electrostatic interaction, and that need to be taken care of with very critically. That needs to be taken care of. So that's why force field is very important. One of such example here is, you can see these are two amino acid residues which are present in the protein structure, and these two amino acid residues should be stably interacting with each other to maintain the integrity of structure. And with the force field, basically you can see how different force field treats it as a differently, and it, because of that different treatment, it changes the conformation of amino acid residues inside the protein structure. And that's the reason why you need to always consider the force field very critically. You need to look for all those interactions, whether it is in the right conformation or not. So that's why I suggested to use the best force field, which for Schrodinger it is OPLS3 force field, which is available with you. So that you need to think of. So other than that, one of the method is loop sampling method. So for loops also, we need to have very good type of method available in the software to treat all those loops. Like loops are basically distorted structure, so it might have various conformations present in it. So you need to sample all those conformation, and loop can be extended from, let's say, 3 amino acid residue to 5 or 10 or 15 or 20 amino acid residue longer loop, and one need to treat those loop structures in very exhaustive manner. So you can see here not only that particular loop structure, but there is implicit solvation method which is also reliable. Because of this environmental effect only, loop gets fold into the protein structure, and for that you need to consider that environmental factor while building your homology model. For example, here you can see the protein is immersed in the water, which is explicit water, but because we need our result faster, so we can't consider it as a explicit water. So we have a specific option called implicit solvation model, in which it considers that environmental factor of that particular water which is present around the protein, and because of this environmental factor, your loop gets fold inside the protein structure, or it gets stable inside the protein structure. So one of the study, according to that study, the statistics, they have compared various loop length and number of cases in which, for example, here the loop length was 14, and in such way there are 36 various protein structures they have studied, and even out of that they came to know about average backbone RMSD for this particular loop with respect to the experimentally determined structure is 0.6, which is very close to the exact same confirmation. Means software is able to generate the exact loop confirmation as of reported by the experimentalist. Even the loop length is like 14 amino acid residue long, and in this way they have performed various loop length like up to even 20 amino acid residue, and they have seen like even for longer loops it has the RMSD value with respect to the experimentally determined structure is 1.5. So that's very critical in case of homology modeling. One need to have that reliability in the loop sampling. So that's why I always say like Schrodinger is predicting a better structure out of all available information. So one need to use that refined loop option to generate a various loop samples, sampling of the loop. So here I have listed there are various challenges which you come across while doing homology modeling that we should not ignore. We always need to check all those things whether you are facing any challenge in doing it. So for example, as I said, like structural alignment is it should be accurate enough and template should be suitable. So if you have high sequence identity and similarity, then most of the things get solved. One don't need to worry about the homology model which is being built. So that's why you always need to look for the very high sequence identical template structure. So if you find it, it all solves the thing. But if it is having less sequence identity or less sequence similarity, then you need to intervene in between for sequence alignment, or while doing homology modeling you need to check iteratively all of your structures, like which structure give you the best, you need to refine it. So it needs some human intervention in case of low sequence identity, so that you need to consider. And because it is all, so one don't need to worry about it because it's a very routine problem in case of homology modeling, because it is all depending on the whatever experimental data available with you, like a template structure which is available in protein data bank. So so sequence alignment and suitable template is one of the biggest challenge which makes your life easy or difficult based on the high sequence of identity and low sequence identity. But even once you build the model, you need to perform model refinement. For that we have various options like side chain confirmations you need to take care of. You need to consider loop confirmations or binding site confirmation. So these are the things after building a model one need to look into it, and you need to see whether on all those parameters your model is good enough. And there are various type of common errors which could happen inside while building a homology model, like rotamer packing or very distortion present in the loop, or which type of loops it looks like, and all those things comes because of that misalignment during sequence alignment. So that's the reason why you always need to look into sequence alignment and check whether it is correct or not. And as I said, like it might happen if you choose a wrong template structure. If you are choosing correct template structure with high sequence identity, then of course these things doesn't come into picture because it will be already taken care of. And there are various based on identity there are limiting factors, like for example if you are dealing with more than 75% of identity, then it will definitely give you good homology model out of it with high speed and high accuracy. But let's say if you are dealing with sequence identity up to less than 75%, let's say 50 to 75%, then you need to do little bit tweaking in alignment, and you also need to go for loop modeling, which might consumes little bit of time. But if it is less than that, like 30 to 50%, then of course alignment becomes very crucial and human intervention becomes very critical. And again, like if it is less than 30, then one need to think very critically that whether it is giving me a good model or not. You need to check at each and every point, and you need to reiterate the same process again and again to get the best homology model out of it. So even once you get the homology model out of it, there are many such parameters through which you need to confirm whether your model is good enough or not. But with Schrodinger, as I always say, life becomes easy. So you will have a single click option called protein quality and reliability report, through which you can consider all those parameters which you need to consider. For example, Ramachandran plot you can have in just single click. You can have various type of deviations like bond angle deviation, bond length deviation, steric clashes. All those things you can see it in a very handy tool in just single click. Or even if you want to see all things holistically, you will have protein reliability report option, in which all those things are being shown in just one view. For example, here wherever you find green color means it's there is no problem with this particular parameter. For example, here it says green color with PDB resolution, means it says it is okay. Or binding site packing, it says it is okay. Protein packing also, it says it is okay. But wherever you find red color means you need to take care of those things. For example, here red color missing loop option is there. It means there are some loops which are missing in the protein structure. As the size of this red color increases means there are some serious problems occurring inside the protein structure. For example, here in particular case steric clashes, and it's a big circle in red color, means there are many steric clashes present in the protein structure that you need to take care of. So how to take care of it is simple. By performing energy minimization, you might get rid of steric clashes. So this is the way holistically one can have a single picture out of for your protein structure validation, and your goal is to improve this all properties in the protein structure. So as I always said, like if you at the end, if whatever model you get, it is not very straightforward process. You need to reiterate the process in case of complications. For example, if you don't have suitable template structure, if you have low sequence identity, then yes, you need to reiterate the process. You need to refine your model and check for the parameters. You need to again refine the model and check for the parameters, and in this way afterwards finally you will get the good model out of it. So always remember, computer aided drug designing is always a iterative process, so never get frustrated out of it. Sometime if you are lucky enough, you get easy answers. Sometime it might take some time to get the answer, but one thing is for sure, if you do it critically, then success is always yours. So with this point, I would like to acknowledge my team who are working in Bangalore round the clock to solve your all issues, and I'm thankful to all of my team for your who are supporting you in all possible ways. So with this, I conclude the session and discussion on our homology model generation. I thank you to all of you for joining us and listen the lecture carefully. And at the end, I would like to open the session for questions if you have. Thank you very much.
Thank you, Dr. Prajwal, for the presentation. We have received a few questions. As a reminder, if you have any questions, please add them in the chat window of the YouTube video, and we'll get them added to our list. So the first question is, if X-ray crystal structure are already available for a protein, is it necessary to carry out homology modeling for such protein?
No need. If X-ray crystal structure is available, means somebody has already experimentally solved it, then there is no point in going for predictions based on homology models. You can directly use it for your further studies, but again, as I said, you need to go through protein preparation wizard to resolve all those problems which are present in the structure.
Thank you. The next question is it a good idea to use cryo-EM structure for computational studies?
So yes, but one need to think of, because whenever you are dealing with cryo-EM structure, it has a low resolution like 3.5 to 5 or 15 angstrom resolution, and with that angstrom actually the visibility of atomic density is very less. But you need to think, if you don't have any other option available other than cryo-EM structure, then yes, it is okay to go with it, but you need to refine that structure first using let's say either protein preparation wizard or using molecular dynamics simulations, and get the reliable structure out of it and proceed for further studies. If you have some option of X-ray crystal structure, then that's well and good. So going further with cryo-EM structure, carefully.
Thank you for the answer. So the next question is, can we use a template from two different experimental methods, for example, from X-ray and cryo-EM to build your model?
Absolutely, yes, it is fine until and unless you can manage with the good sequence alignment. Then yes, you can use it from different methods.
Thank you. So the next question is how to choose template protein structure from available different protein. I think the person meant like after the blast search, when we get a lot of hits, how do we choose which one is the right template to use?
Yeah, so for that, here I have mentioned few of the parameters like sequence identity and similarity. Then what is query coverage? It should be maximum. Identity and similarity should be maximum. Then there should not be any missing residues in the template structure. Its resolution should be good. It should be with ligand if possible, and that could be most active ligand. So based on these parameters, you need to decide the best suitable template structure.
Right. So the next question is, what should be the structure format while preparing for homology modeling? Will it be .pdb? I think it means the output.
Yeah, I mean it's fine. Like the structural format should not, you should not bother about it. If you are doing with Schrodinger, then of course it will generate it in a MAE format, which is supported by Schrodinger. So one don't need to worry about it. For protein structure, you can use MAE or PDB format.
Right. So the next question is, what is the algorithm behind Maestro's homology modeling tool?
So algorithm is same. So these are the various steps which software follows in background. So this is what the step-by-step manner it will give you the final homology model out of it. There can be various methods which one can see, like knowledge-based method. In case of high sequence identity or similarity, you can go with knowledge-based methods, or if you believe like knowledge-based method is not working fine with you, one can go for energy-based method, in which a software tries to identify some of the regions in the protein structure based on energetic values. So these are the things which we need to take care of.
Right. So the next question is, while doing the homology modeling, if your protein of interest, the structure is not present in the same organism, can you use the protein structure from a different organism in order to build the homology model? For example, if a protein is present in E. coli, but you're trying to build a homology model for Mycobacterium tuberculosis, for example, can that be done?
Yes, it is fine. If you get a good sequence identity and good sequence similarity, then yes, it is fine with any other organism structure.
Thank you. So if the sequence similarity is less than 80%, how many templates should you use?
So the straightforward answer is always remember one thing. If you have option to go for single template alignment or single template modeling, that's the best way. Don't go for multiple template until and unless you have no choice available. Because whenever multiple templates come into picture, complexity increases and reliability decreases. So if you have a single template available, and the sequence similarity can be even if it is less than 50%, and single template is available, then also it is okay to go with it. So I would say like 80% is very high sequence similarity. So 80% like you don't need to worry about it. So if it is less than 50%, then only you need to think of multiple template homology. Otherwise, no need to even think of it. It's all fine.
Thank you. So the next question is, how do we read Ramachandran plot?
So how do we read Ramachandran plot? So the simple thing is Ramachandran plot, you can see here on the bottom left, it is divided into various regions. Like red color is the most allowed region, then yellow color region is the allowed region, and white color region is disallowed region. So these dots, if you see, this dot represents each amino acid residue backbone torsions. So this dot should not fall in white color region because it is disallowed region. So you would need to get all those dots inside red color or yellow color region. So this is what the best way to read it. But at any given point, if you want to read more about it, so simply type Ramachandran plot in Google and you will get the first link from Wikipedia where it is all explained in details.
Thank you. So the next question is, if we have an apo structure, should we opt for homology modeling?
So if you have a post structure, then structure is there. You can proceed further for further studies like all molecular docking or induced fit docking. So if structure is available, then no need to go for homology modeling.
Right. So the next question is, okay, I'm reading it as it is. Is there any database available to do Ramachandran plot?
So I think question you need to tweak little bit, but you don't need to worry about it. Like it's just a single click, and there is no database for Ramachandran plot. It generates based on the whatever structure available with you. So with Schrodinger, you will have a simple tool to generate it. It's just a single click option.
Right. So the next question is, while doing the homology modeling conventionally, after multiple sequence alignment, we have to build a PSM profile. Does Schrodinger also do that?
What is meant by PSM?
I have no idea. So if Surbi, you could just explain that to us.
It's
I
a
have a feeling it's from some other software.
position specific matrix.
Okay, so it's not only about PSM, but there are a number of factors which are available as with respect to various softwares. Every software has different ways to analyze it. So, I mean, whatever is available with Schrodinger, those are the essential things which you will have. But it is again not limited. You go to any other software, any other web server, you will find some other parameters. But at the end, what is take-home message? Take-home message is you need to fall within the limit. You need to have all things in allowed regions. So there can be Z score, there can be red plot, there can be PSM, there can be PROSA. So there are many such things which are available. So if you again with homology modeling, you always need to think if your model is well good, like it all follows the Ramachandran plot and steric clashes and this protein reliability report parameters, then everything will be taken care of. So you don't need to look for hundreds of such parameters at the end. It is all being covered.
Right. So the next question is there a database available where you can get homology model proteins?
Yeah, there are databases available like SwissProt database where people used to deposit the homology model of protein if it is available. But again, whatever homology model is, it is all predicted structure only. They can be false positives or true negatives type of model. Whatever you get it from such databases, you need to revalidate it. So that's the best possible way. So if you get such model, get it, but don't believe it blindly. Well, revalidate it. If it is good enough for you, then only proceed further. But there are databases available.
So the next question is, how many templates can be used to generate a model?
So
I think it means like the upper limit maybe.
So there is nothing like upper limit. It's all up to you how many templates you want to use. But my suggestion is always there. try to use as less as possible templates. If it is single template, that's the best scenario. If it is 2 template or 3 template, it's fine. If it is more than that, the reliability will be very, very low. But you can use hundreds of template if you want.
Can you please tell us the need of blast analysis, and is that available using Schrodinger suites?
Yes, it is available with Schrodinger suite. So BLAST is nothing but the method to find out best template based on sequence identity or similarity. It's one of the method of alignment of 2 sequences. So it is available with Schrodinger suite.
Right. So the next question is, when we do a 2 template based modeling, what is the importance of RMSD with respect to template in such case? Is there a possibility that the model can have a confirmation in between them?
Yes, so possibility is always there for anything, and the RMSD means always remember RMSD value should be less. It can be up to 0 also, means it should be less. Whatever it is, whatever possible less you will have, that's the best, better thing.
Right. I think you already answered the next question. So when the PDB is available for E. coli instead of Homo sapien, then do we need to go for homology modeling?
Yeah, I mean, which organism protein you want to study. So if PDB structure is available for E. coli and you want to study human protein, then answer is yes, you need to go for homology modeling. So because as organism changes, amino acid residue sequence change, and as sequence change, the structure might change. So you cannot use E. coli structures to study human protein. You need to build a homology model.
Right. So the next question is, which is the better method, knowledge based or energy based?
It depends on your requirement. So whatever information available with you. So knowledge based method has own significance when you have high sequence identity or high sequence similarity. Knowledge based method works better. But if you have low sequence identity and similarity, in that case, you need to go for energy based method.
Okay, so the next question is, if in the protein reliability report, many of the parameters are not good, then what are the other options to go for except to change the template?
Yeah, so what are the other ways to change it? You can perform loop refinement, you can perform energy minimization, you can perform rotamer search, you can perform side chain search. So these are the various things which are available with us to refine your model, to get your protein reliability report better.
What should we do if we do not get any protein template for the homology modeling?
So if you don't get any protein template, then I would not suggest you to go for such a homology modeling method, because it will not generate any type of reliable model. It's all template based homology modeling, so that's why template should be there.
Right. What is forbidden region in Ramachandran plot?
So I mean, there are a few regions which are, for example, you can see amino acid residues like prolines and glycines. So those are the amino acid residues are kind of exceptions to the Ramachandran plot. So they can fall within a disallowed region, and there should not be any worry out of it. So it will be fine.
Right. I think they mean DFT.
Shelvi, I'm sorry. I think I missed. There was voice cut in between.
Sorry. Can you hear me now?
Yeah, it's fine.
Okay, sorry. Um, So the question is the density factor calculation compulsory for a molecule in silico study? I think they mean DFT calculation.
Maybe, I'm not sure.
Okay. Okay. Let's just move on. Okay. How chiral molecules respond to the target protein? Is there any impact or special arrangement in homology model?
So, no, I think that question needs to be refined. So you can see even there are various chiral molecules available that can have huge impact on biological activity. Like R isomer is most active and S isomer is completely inactive. So there are such examples of drug molecules. So you always need to consider such parameters. And with respect to homology model, so yes, according to the stereoisomer, which is active, protein acquires a particular shape in binding pocket, and not only that, it is vice versa also, like because of that shape of binding pocket, only that particular chiral molecule can fit inside it. So yes, it has impact. One need to take care of it also.
Right. The next question is there any successful story with homology modeling?
There are n number of studies available. You just need to type it in a Google search and you will find a lot of papers available on it.
So the next question is there an application where we can find the pockets in the protein?
Yes, we have seen it in previous lectures. There is a tool called sitemap through which we can identify the various pockets inside the protein structure.
So the next question is if the DFG in state is available for a kinase, then for type 2 inhibitor identification, what will be better? A homology model for the inactive or outstate or induced fit docking would help.
So if you will have option to build a homology model for active state based on the active template, then that's the best possible way. If you by chance don't have such option available, like active state template is not available, then next way is induced fit docking.
Okay, I think we've come to the last question. Is there a basic course where we can learn homology modeling and MDMS?
So it's here, you are on the right platform. So this is what is the basics of homology modeling, and in consecutive lectures you will see how to do it actually in the software. And for MDS, the next session will be on MDS. Just stay tuned. So just after 5 minutes it will start, and you will have the basics of molecular dynamic simulations also.
Okay, sorry, spoke too soon. So there's a clarification by the person who asked the question before. So they're just asking if DST is compulsory to go for the in silico study.
DFT.
Yeah.
So, I mean, it all depends on you, what problem you want to address. So always remember there are n number of studies, n number of methods which are available to solve the problem, but you need to ask yourself what is the problem which you want to address, what is the problem you want to solve. Whether is it possible to solve it through this particular method, or I need to look for any other method. So if you will have answer for it, then yes, you can use molecular mechanics method, you can use DFT methods, you can use QM methods. So all methods are available. You need to think of the problem which you want to address.
Right. So the next question is, do all these tools like pocket prediction, target preparation, and homology modeling, etc., do they also work on RNA targets?
Yeah, as such, they should work, but with sitemap, it is specifically optimized for the protein amino acid residues. So it will give you the best result for protein. For DNAs, for RNAs, you need to think like RNAs and DNAs has double helical or single helical structure, and it has very specific grooves, which are my major groove and minor grooves. So in such cases, like it is very unlikely that you need to go for such pocket prediction studies, because it's all definite things which are present. But in case of protein, it's very uneven, so you need such tools.
Right. There's one last question. How to calculate volume for active sites in the homology model without the ligand? Is there an option in sitemap?
Yes, you just need to open the sitemap and run it. It will give you all those parameters like size, volume, other parameters. It's all there.
Thank you, Dr. Prajwal, for answering those questions and for your presentation. I think it was very popular since we received so many questions. We are just in time for the second presentation of the day. So the second presentation is a molecular dynamics simulation theory and analysis by Dr. Kaushik. Yes, so I'll just pass over the controls to you. I can see your screen.
Yeah, thank you very much, Shelvia, and thank you very much to all of the participants. And over to you, Dr. Kaushik.
Thanks, Prajwal, and thanks, Shelvia. Can you hear me?
Yes, we can hear you.
Perfect. Thank you. Hi, everyone. I'll be talking about molecular dynamics simulations and how you can apply molecular dynamics in drug discovery. If you have any questions, continue to post your questions in the YouTube chat, and we'll get back to the questions after we finish the talk. All right, let's get started. So why do you need to do molecular dynamics? Last week, we looked at so many different concepts of docking. Our goal is to study how the drug interacts with the receptor. So here I have a receptor, you have a ligand, and you want to study how this ligand interacts with the receptor and how do you stop the activity of a protein by using a ligand. And for that purpose, last week we looked at so many different topics, from introduction to computational drug design, where we looked at structure-based drug design methods. We looked at ligand-based, we just introduced ligand-based drug design methods. We'll go into details of ligand-based drug design methods tomorrow. We also looked at target structure understanding. How do you select a structure from X-ray? How do you select a structure using NMR, or if the structure is inhibitor bound? We looked at all of these things last week. We also looked at ligand library for simulation, where we looked at different databases that you can use to prepare your ligands. We have Zinc databases, we have PubChem, we have other databases that you can use. And so you have all of these options. And we also looked at protein preparation and ligand preparation for simulation, for docking, not for simulation, where if you have missing residues in your protein, how do you fix the residues? How do you generate different tautomers for ligands? How do you generate different potential states for your protein? We looked at all of those things last week. And after that, we also tried to identify ligand binding sites using sitemap. All of these were covered last week. We also covered the theory, principles, and methods of molecular docking using GLIDE. We looked at high throughput virtual screening, HTVS. We also looked at docking using single precision mode. We also looked at docking using extra precision mode. We saw all of these things. And we also had a session on virtual screening and how do you prioritize the molecules? How do you identify a hit molecule? How do you identify a lead molecule? We looked at all of these things. And finally, we also had a session on flexible docking and covalent docking, where we looked at induced fit docking and also a tool called covalent docking. So after last week's sessions and also with today's session on homology modeling in the morning, probably all of you are confident of doing how to do docking. So if you know how to do docking, then why do you need to do molecular dynamics? Well, docking, when you do docking, if you see this particular video here, if you look at this, the protein is fixed and you only change the ligand. However, in reality, everything is moving. We are moving, when we are moving, the proteins in our body are working, and they're working to work. They have to move, and the ligands have to move. They have to go through the different parts of the body to go to the binding pocket. And so everything is moving all the time. And so if you have a rigid receptor like here, it's not an accurate representation of what is happening in our body at all times. And for that reason, you always have to include some sort of dynamics in your calculations. And since docking does not do it, last week we also covered induced fit docking. So just to recap again, docking is very simple. The protein is rigid, only the ligand is moving. And because of this particular, because the protein is not moving, the calculations are really fast, they are really quick, and it's very, very cheap to do the calculation. So if you have like 1 lakh compounds, you can do docking and you can get your answer in 1 or 2 days for those 1 lakh compounds. It's also very easy to score it because the protein is not moving. So all of these calculations are really fast. But after you do docking, let's say you have like 100 molecules or 10 molecules that you finalize that are really good ones. And for those ones, you can try to do induced fit docking. In induced fit docking, you have the binding pocket is flexible, only the binding pocket. The protein is not flexible. And the ligand is also flexible. And because the binding pocket is flexible, the calculations are slow. And it's computationally expensive compared to docking. And you also have different conformations of ligands and binding site residues that are being explored in induced fit docking. However, even after you do induced fit docking, the binding pocket might be flexible, but in reality, the whole protein is always flexible. And for that reason, you always want to go for molecular dynamic simulations as an end step for your molecular docking process. In molecular dynamic simulations, the whole protein is flexible. The ligand is also flexible. And because all of these atoms are flexible and all of these atoms are moving, the calculations are very slow, and it's very, very expensive to do it computationally. And you have to explore different conformations of ligands and proteins, so you have to do all of these things. For that reason, molecular dynamic simulations are the last step that you usually do when you're trying to identify a ligand molecule to interact with your protein. So just to recap, docking is fast and simple. Induced fit docking has slightly more flexibility. It's computationally slightly more expensive, but MD simulations are the gold standard for evaluating your ligand poses. So just to add to that, what you can do in MD simulations is that you can try to understand the stability of a drug binding. In docking, you know that the ligand interacts with the protein, but you don't know how well the ligand interacts with the protein. So for example, if you look at this video, if it's an MD simulation of a protein, which is shown as a surface representation in white, gray color, and the ligand is shown in orange here, and the ligand is interacting with the protein. And you can see that in this particular video, if this ligand is present in the binding pocket, it is not leaving the binding pocket, then it indicates that the drug is stable. And because it is a really expensive method, you only want to do MD simulations for like 10 ligands or 20 ligands. You don't want to do it for 1 lakh compounds. And so you have to initially get your number of molecules from 1 lakh to 10 or 20 using docking or induced fit docking. And after you have those two, narrow it down to 10, 20 molecules, then you can do MD simulations. And MD simulation is also useful for identifying ligands with similar docking scores. So you can differentiate between ligands that have similar docking scores. So let's say you do docking using GLIDE, and then you get like 4 different ligands that have similar binding scores, like -11 to -10. You don't know which one of these is the best one. They're all in the same range. GLIDE is going to tell you that it's -11 is the best one, but you can only be confident of your answer only after you do molecular dynamic simulations. And when you do the molecular dynamic simulations, you can identify which of the ligands is actually stable in here because the protein is moving, the ligand is moving, the interactions might change between the protein and the ligand. And so all of these scores that you see of -11, -10 are refined by better picture of what is happening in reality. And so MD simulations can be used to differentiate between the ligands that have similar scores. And so that's why you always have to do MD simulations after you're docking. It's always recommended. And if you want to get your paper published, then they always recommend you to do MD simulations for your final docking poses. So the next question is, how does MD simulations work? The basic idea is very simple. What you have to do is you have to somehow find a way to move the atoms. So you have to move the atoms in the protein, you have to move the atoms in the ligand, and you have to do them simultaneously. So you can state the interactions as a function of time. And so let's just see how the basics of MD simulation works. Let's take a hypothetical molecule. So here I have a simple molecule. The same concepts apply for a bigger molecule like a protein. The same concepts apply for DNA, RNA. The same concepts apply even if you have a membrane bound protein. So let's just look at the simple molecule and try to understand how molecular dynamics works for the simple molecule. So if you look at this molecule, it has 7 carbons, 1, 2, 3, 4, 5, 6, and 7. It also has 2 oxygens here. It's a carboxyl group here. And you also have a NH3 plus atom here. So you have all of these atoms present in this molecule. And when you try to do MD simulations, you first need to identify all the interactions that are present in this particular molecule. So for example, if you look at this particular molecule, you have a bond between atoms 2 and 3. You also have a bond between atoms 1 and 2. You have a bond between 3 and 4, 4 and 5, 5 and 7, 5 and 6, 7 and oxygen this one, 7 and oxygen minus, 3 and nitrogen here. So all of these bonds are, for all of these bonds, you have a simple equation which looks like this. You don't have to memorize these equations. All of these things are taken care of with the software. I'm just introducing the concepts here so that you're aware of it. So you have all of these bonds, which are like 6 of them plus 2 plus 1, 9 total bonds. And so for each of these bonds, you have different parameters that you can have. All of these things are already built into the software. You don't have to worry about it. You just use this and you try to check this, calculate the bond energy for each of these bonds. Once you find the bond energies, the next thing that you have to calculate is the angles. So angles are defined for every 3 atoms. And so you have an angle between 1, 2, and 3. You have an angle between 2, 3, and 4, 3, 4, and 5, and so on. And you get the basic gist of it. And so for all of these angles, you also calculate the energy for each of these angles. And the calculation is based on this equation. Again, you don't have to memorize it. It's good to be aware of it. And you don't need to know it if you have to do simulations. And the next thing that you can also, that is also being calculated is if you have 4 atoms, like you have 1, 2, 3, and 4. And so here I have a simple molecule, butane. And if you remember from your intermediate chemistry, you remember that butane has different conformations. You can have a trans conformation. You can have a cis conformation. The cis conformation is less stable compared to the trans conformation. And for modeling this type of interactions where one conformation is more stable than the other conformation, you also have a term called dihedrals. And there's the formula for calculating the dihedral energy. And you don't have to memorize this thing again. And these things are taken care of the software. I'm just introducing concepts here again. And so it's not just done for these 4 atoms. You also do it for 2, 3, 4, 5. You do it for 3, 4, 5, 6. You do it for all of these atoms that have an interaction like nitrogen 3, 4, and 5, 3, 4, 5, and 7. You can do all of these calculations. But if you look at this particular molecule here, you also have a few interactions that are non-bonded. So for example, there is no bond between nitrogen and oxygen here. And you also have to estimate the interaction between nitrogen and oxygen. So how do you do that? So for calculating non-bonded interactions, there are 2 different terms. One is called electrostatic. It's a simple equation that you learned in your intermediate physics, where you have the charge of nitrogen, the charge of oxygen, and you multiply the 2 charges and divide it by the distance. Sorry, this should be Rij, not Rij square. So you calculate the energy, electrostatic energy here. And you can also have these Lennard-Jones interactions, which prevent the 2 atoms from coming very close to each other to avoid steric clashes. So you have all of these equations that are there. So we looked at these 3, and then we looked at these 2 here. And all of these interactions you calculate for every pair of atoms in your protein, every possible setup that you have. If you have bonded interactions, you calculate the bonded energies. If you have angle, then you calculate all the angles. If you have dihedral, you calculate all the dihedrals. You also calculate all the charge-charge interactions. You also calculate all the Lennard-Jones interactions. And once you calculate all of these interactions, then you have this master equation. And this whole equation is together called a force field. Again, let me emphasize this. You don't have to know all of these equations. You just have to know that all of these things contribute to the force field. And there are so many different force fields that are available right now. And Schrodinger uses OPLS3E. The advantage of using OPLS3E is that it's not just that you have these parameters, these equations for the protein or for the DNA or for the RNA. It also has parameters for 95% of the chemical space, which includes all the ligands that you can think of, most of the ligands that you can think of, all the DNA, all the protein, all the different nucleotides, different amino acids. We can also think of modified amino acids like phosphorylated amino acids. It has the parameters for all of these different atoms and residues and molecules. And because you have all of these parameters in one single force field, it's very easy to identify the interaction between, let's say, a protein and a ligand, or DNA and protein, or protein and RNA. You can do all of these interactions, study all of these interactions. All of these are pre-calculated. You don't have to worry about any of these things. They're all fit into the software. All you have to do is just use the software and do the calculations. So once you know how the forces are being calculated for each of these atoms, once you know like how to do the bookkeeping, how many bonds are there, how many angles are there, how many dihedrals are there, how many charge interactions are there, how many Lagrange interactions are there, the next thing that you have to do is you have to calculate the force acting on each atom that is present in your system. For example, here I have 3 atoms instead of the 9 atoms that I had before. So you have atom 1, atom 2, atom 3. For each of these atoms, you calculate the force coming from atom 2, atom 3, and you calculate all the forces on atom 1 here. So it's just simple, basic intermediate algebra, vector algebra, so you can do all of these calculations. And once you have the forces, you calculate the acceleration just by dividing the force with the mass. This is the same equation that you looked at so many times when you're doing intermediate. F equal to ma, we just rearrange this to be A equal to F over m. And this acceleration is for each atom, this force is for each atom, this mass is for the same particular atom. So once you calculate the acceleration from the force, the next thing is if you know acceleration, then you can calculate velocity by using this equation where acceleration is equal to velocity over time. And once you have the velocity, you can calculate the position of the atom by using this equation. If you know velocity, you can just use this equation R over T, and you multiply the velocity with the time, you get the position of the atom. So why do we have to do this? Because in molecular dynamics, you want to move these atoms from one place to the second place. For example, here you have the black atoms, which is the initial position here in black. And the same atom is moved to this gray position here. So atom 1 moves to the gray area here. Atom 2 moves to the gray area here. Atom 3 moves to the gray area here. So all of these things have to be moved. And to move them, again, let me just repeat this. You first have to find the forces using the force field. Then you calculate the acceleration. And once you have the acceleration, you have the velocity. And once you have the velocity, you can have the positions. And you just have to calculate this. So if you look at this particular set of equations here, you don't have to memorize anything. It's just very simple here. All you're trying to do is R of t, which indicates the position of, let's say, atom 2 at time t. You just say what is the position going to be after a time step of delta t. And delta t indicates a time difference. So I have the black circle here at time t, and I have the gray circle here at time t plus delta t. And R is just like a combination of the change in x direction, the change in y direction, the change in z direction. And you just add these smoothest atoms for a small time step. And the time step is typically 1 to 5 femtoseconds, depending on your different ways of calculating it. You don't have to worry about any of these things. You just have to, if it's good to be aware of them, you don't have to memorize it. The software does all of these calculations in the background. The only thing that you have to worry about is how many times do you have to run this calculation? So if you have 1 femtosecond, then if you want to do 1 picosecond calculations, simulations, then you have to do this, repeat this calculation 1,000 times. If you have to run it for 1 nanosecond, then you have to repeat this calculation 1,000,000 times, like that is 10 lakh times. And if you want to do this calculation for a microsecond, then you have to do it for 100 crore times. And because you have to do it for 10 lakh times or 100 crore times, this method is computationally very expensive compared to the other methods. And so again, you don't have to worry about how long it takes. Like for the study that you're trying to do right now, if you just want to identify how long it takes for to know if your ligand is having a stable interaction with your protein, you can just do the calculation for a few nanoseconds, like 50 nanoseconds, that's sufficient. And depending on, if you look at this chart, there are different things that you can study in different time scales. If you want to look at the bond vibrations, that is each individual bond length, then you have to do the simulation for a few femtoseconds. If you want to look at side chain rotamers, then you have to do the calculation for a few picoseconds to nanoseconds. And if you want to look at large scale loop motion where your loop is changing, then you have to do the calculation for a few nanoseconds to a few microseconds. And if you want to look at ligand binding and unbinding, that is, you don't know where the ligand is, you have the ligand in outside the protein, and if you want to understand how the ligand goes into the protein, then you have to do the calculation for microseconds to milliseconds. But in your case, you already know that the ligand is docked inside the protein, and all you're trying to do is try to understand the stability of the ligand of the protein. And for that, all you have to do is do simulations for 50 nanoseconds or 100 nanoseconds. And you might think that, if you have to do it for 50 nanoseconds, then you have to do the calculation 50 lakh times, which is a lot of times. But with GPUs these days, you can easily do calculations of 100 nanoseconds per day on a standard size protein. So if you have to do for 50 nanoseconds, then all you need to do is you have one GPU, and you can do the simulation for half a day, and you can identify if your ligand has a stable interaction with the protein or not. So it is really easy to do this. So just to recap, all you have to do is, use the software. The software is going to calculate the forces for you in your protein and ligand. It's also going to move the atoms for you. And all you have to do is just tell the software how long you have to do the calculation for. So let's look at how you can use Schrodinger to do the MD simulations. You can use different softwares to do it, but using Schrodinger, it's very, very easy to do the calculations to set the MD simulations. You first have to get your protein from the PDB. And once you have your protein, you use the protein preparation wizard, which we looked at last week. And we're also going to have demonstrations next week on how to do these calculations. And you just have to make sure that your protein is complete. It has all the hydrogens. It has every missing residue is covered. You don't have any missing residues in your loop. The side chains are in optimal position. You prepare your protein. Once you prepare your protein, you have to build a system for doing the simulations. And it is easy to do it. I'll go into details in the next few slides. Where you can set up your membrane if you have a membrane. You can set up your water if you have water. And you can set up, I can add ions to your system. You can do all of these things using System Builder. And once you do the system building, then you go to MD simulation, where you do the simulation here, and you have the protein moving, you have the ligand moving, you can study the interaction between the two of them. And once you do the simulation, you can analyze your simulation. And all of these things are single clicks. It's very easy to do it using Schrodinger. I'll just show them in a few slides, in the next few slides, how easy it is to do the simulation, and also how easy it is to do the analysis of the simulation. And there's just a brief workflow of how you can do MD simulation in Schrodinger. So let's just look at how to do the system builder in the next slide. So Desmond is the molecular package for doing the MD simulations in Schrodinger. It is the fastest MD engine compared to other softwares. So Aemberg, Romax are slower compared to Desmond. And if you look at this particular interface, it's all mouse clicks. You don't have to do any commands. You don't have to type any commands. It's just, you just click on different things. It's very easy to build your system. And if you look at this particular image here, I have the protein, which I'm showing you in cartoon representation here. There's a ligand, which is not so clearly visible. You have these red dots here. These red dots are all water molecules. Why do you need to have water molecules? You need to have water molecules because most of our body is 70% water. So you have to be as close to reality as possible. So you want to have all of that water, so which is indicated here in a box. And you can also add ions. So it's very easy to build it. All you have to do is just click on these different options that you have. For example, if you want to choose different water models, you have different water models here. You have SPC, T3P, T4P. You also have octanol or methanol if you want to have co-solvents in your system. You can add all of these things. It's very easy to choose it. All you have to do is just click on this box here and just drop down and look at these different options that you have. And once you select your water model, you can also use water models outside these things with external scripts, which you don't have to worry about. It's just easy to use these options here. TIP3P is a good model to start with. You can also use TIP4P EW, and you you can use any one of these water models for you to have a decent simulation. Once you select the water model, the next thing that you have to do is you define what type of box you want. Do you want a box that is cube-like? Do you want a box that is different shaped? You can set your box dimensions here. You can set a buffer. You can do all of these things. It's all mouse clicks. It's very easy to set up. And it's also very easy to choose the box dimensions. Once you do both of these things, you can also set up a membrane. If you have a membrane protein like GPCR, which is transmembrane protein, for example, all you have to do is just click on this option here, and you can set up different types of membranes like POPC or different membrane gray, which is shown here. You can set up all of these things with just simple mouse clicks. And once you set up your membrane, if you have membrane protein, then you can also add ions to your system. It's very easy to add ions. It automatically adds ions depending on the charge of your protein. You have all of these options to add salt as well. So all of these things are mouse clicks. You don't have to worry about setting up through command line. It's just very easy to use it. Click, click, click. And like with 3 or 4 clicks, you can set up your whole system. And once you set up your system, the next thing you have to do is you have to start the simulation. And to start the simulation, there are a few things that you have to consider. It's, again, very, very easy to start the simulation using Schrodinger. The first thing you have to do is you have to load your molecule that is there. So here you can see that, it tells you how many number of atoms you have in your system. You have to move all of those atoms. You have to identify how long you have to move the atoms for. So you can move them for 10 nanoseconds, or you can move them for 100 nanoseconds. You can move them for a few picoseconds too, depending on what you're trying to study. And setting time is just like you just have to enter your time. It just does the calculation here automatically. So it's very easy to do that. And once you set up your time, the next thing you have to do is choose an ensemble. Again, these are all different options that are there. You don't have to worry about these different options. Just use the default options and you're good to go. But if you want to have more control, you have these different ensembles that are supported. You have a NVE ensemble, which is normal volume and energy. NVT ensemble is normal volume and temperature. NPT is normal pressure and temperature. These 2 options are useful if you have a protein in solvent and ligand in solvent. If you have a membrane protein, then you might want to use NPAT. A stands for area, or NP gamma T, where gamma stands for surface tension. If you have membranes, then you want to use these 2 ensembles. If you do not have a membrane, then you can just use these 2. And all of these things are default options. Just use NPT and you're good to go. So once you do that, you also have a few advanced options where you can choose a thermostat or a barostat. Why do you want to have different temperatures? Well, most of us exist at the human body is usually at 298 Kelvin or 300 Kelvin. 37 degrees centigrade, and that's a normal temperature. But there are some bacteria that exist at really high temperatures, like 80 degrees centigrade. You can try to set the temperature using these advanced options. You can also try to choose different thermostat and barostat, like Nosehover thermostat or Langevin thermostat. There are different options that you can choose. You don't have to know any of these things. The default options work well, and it's just for people who want to be aware of these different options that you have that I'm telling you these options. So just to recap, we looked at how to build a system, how to add ions, how to add water, how to add membrane, and how to choose the time that you want another simulation for, which ensemble that you want to use, and how do you do different set up different thermostats or barostats, and all of these options. So once you start your simulation, the next thing you have to do is you have to look at the trajectory. So here I have the protein, I have a ligand here, I have these different water molecules here that is shown here. And you can see that in this particular video, all of these atoms are moving, the protein is moving, the ligand is moving, the water molecules are moving. And here I just remove the water molecules. You can look at this particular interaction of the ligand with the protein. You can also like zoom into the interactions. You can look at exactly what is happening to the ligand and how it is interacting with the protein. You can see this ligand atoms moving here. You can see the dashed yellow lines, which indicate the interaction with the protein. We have all of these things that you can look at. You can zoom out a bit. You can look at the interactions between the ligand and the amino acids here in more detail. See here, you can see the interaction with leucine, lysine. You can, when the ligand, you can see all of these interactions. And so all of these things can be done using a MD simulation. Just to show you the video again. So you start with your protein and ligand in a water box. You do your simulation. And as simulation is proceeding, you look at the different interactions between the ligand and the protein. And so you can also see so many different things here. It's just so much more convenient to look at all of these interactions between the ligand and the protein and understand how stable each of these interactions is. Now, when you're trying to look at the interactions like this, it's very difficult for you to identify what interactions are happening. It's moving so fast. I can't really identify any of the interactions. I don't know which one is stable. I see these lines forming and disappearing. I have no idea what is happening here. So is there a better way to look at these interactions? Can I be more confident on what are the most stable interactions? How is the protein interacting with the ligand? Can I do more analysis? So just to do that, there is a separate tool in Schrodinger, which is called the simulation interaction diagram. It's very easy to do the analysis. All you have to do is just select all of these boxes, click select all, and then you click run. It's going to do the analysis. It takes around 5 minutes to do the analysis. And after you do the analysis, you can just look at your results. So what do you want to look at? We already did docking in the previous sessions. We know how docking works. We know that the ligand is interacting with the protein. What we want to do is we want to look at the ligand stability. So if you look at this particular video here, you have the protein and you have the ligand, and the ligand is interacting with the protein, and you want to look at how stable the ligand is. And so one way to understand how stable the ligand is to look at RMSD plots. The RMSD tells you how much the ligand is moving from its initial position and how much is the protein deviating from its initial position. So if you look at this particular graph here, you have on the x-axis, you have time we did the simulation 5 nanoseconds. And on the y-axis, you have RMSD. In blue, you have the protein RMSD. The protein is moving. You can see that the RMSD is changing through as simulation is going on, which indicates that the protein is moving. And if you look at the red one, which is the ligand RMSD, you can see that the ligand RMSD is less than 2 angstrom for the entirety of the simulation. This indicates that the ligand is stable inside the binding pocket of the protein. So you did your docking and you're trying to do the simulation, and the ligand is not leaving the binding pocket. This indicates that it's a strong interaction with the protein. And it's a good inhibitor for the protein for whatever you're trying to inhibit or whatever you're trying to study using the ligand. And you can also you can look at the stability of your ligand with the protein, or you can also look at the individual interactions, like what exactly is happening in this between the ligand and the protein. And if you want to look at these individual interactions, it's very difficult to look at it in a video form here. So there are tools that are there. And again, just using the simulation interaction diagram, which we looked at before, you can understand the individual interactions. So you can get like really clear plots like the one that is shown here, where there are 2 different plots. I will tell you each one individually. In the bottom plot here, what you have on the y-axis is the amino acids on the protein, on the receptor. On the x-axis, you have time. And the color indicates the number of contacts you have between the amino acid and the ligand. For example, if you have a darker color red, then it indicates that this particular amino acid has a has a higher number of contacts with the ligand. Like for example, it has more than 4 contacts with the ligand, or 3 or 4 contacts with the ligand here. And these amino acids have contacts forming, and then you see that it's a white region here, which means that the contact is disappearing. And so it forms and it doesn't form. So it indicates that this particular amino acid might not be important for the stability of the ligand. And for example, if you look at these amino acids here, these are completely white, which indicates that these amino acids do not interact with the ligand at all. And so you can look at all of these contacts between the ligand and the protein in a clear graph representation. You can also look at it in terms of the type of interaction that is forming here. So there are different colors here for each amino acid. The x-axis indicates the different amino acids in the protein. The y-axis indicates the fraction of contacts that you have with the ligand for each of these amino acids. The color code is like if it's green, then it's a hydrogen bond interaction. If it is blue, then it's a water bridge. If it is red, which is not shown here, it's an ionic interaction. If it is purple, it's a hydrophobic interaction. You have all of these interactions that you can study between the protein and the ligand using this particular diagrams here. All these plots are generated automatically for you. And it might be a bit difficult for you to interpret this. You can also have a ligand interaction diagram, which is automatically generated for you, where you might have looked at this from the presentations last week, where this one tells you the interaction between the ligand and the amino acids on the protein that you have, and how. So when you use this ligand interaction diagram for a simulation, it not only tells you what interactions you have between the amino acid and the protein and the ligand, it also tells you how long the interaction is present for. For example, if you look at the interaction between this NH group and this glutamate, it tells you that this interaction is present for 100% of the simulation, or this interaction is present for 95% of the simulation. So you have these analyses that you can use to do after you do your simulation, which tells you if your ligand is stable with the protein and why is it stable. And so if you have like 4 or 5 different ligands, you can understand like why a particular ligand is more stable compared to other ligand just by looking at these diagrams here. And so it tells you like, if you have like 4 or 5 different ligands with a similar score, you can identify why one particular ligand is more stable than the other one just by looking at these diagrams. And this is from a ligand perspective. So you can look at the interaction between ligand and protein from a ligand perspective. You can also look at the protein fluctuations. So if you have a part of the protein that is interacting with the ligand, then those parts will be moving less compared to the rest of the protein. And so you can measure those particular fluctuations too. So if a particular amino acid in the protein has interaction with the ligand, it's going to move less. So if you look at this particular plot here, you have the residues on the protein on the x-axis, and you have the fluctuations on the y-axis. And if you look at this particular plot, you have high fluctuations here, you have low fluctuations for some of them, and you have high fluctuations, low fluctuations, and all of this is changing for each amino acid. And if you look at this particular plot here, if you look at these particular green lines here, this indicates that these particular amino acids are interacting with the ligand, and you can see that the ones that interact with the ligand have lower fluctuations compared to the ones that do not interact with the ligand. So this tells you like which part of the protein is interacting with the ligand. So you can try to modify your protein if you want to, if you know what you're trying to do, or you can try to modify your ligand, change the functional groups to understand why certain amino acids are interacting with the ligand and why certain other amino acids are not interacting with the ligand. So you can do all of these analysis. And these are just some of them. There are like a lot more, many more analysis that you can do here. You can also study how a ligand goes into the protein and leaves the protein, binding, unbinding. You can do all of those studies. There are other methods of molecular dynamics that you can use, like metadynamics or replication molecular dynamics, or you can also use free energy perturbation. There are these different methods that are available for you, which you can use to calculate your simulations in a faster way, in a better way, in a more accurate way. So all of these options are there. I'm not going to details of these things. It's just good to be aware of that there are some options that you can use. And lastly, I would like to thank the team that all of us are here. So this is our entire team. And I'll be happy to take any questions. And if I'm not able to answer any of the questions, you can always email me the questions at kaushik@schrodinger.com, or you can always call me on my mobile number that is shown here. And I'll be happy to take any questions that are there.
Thank you, Dr. Kaushik, for that wonderful presentation. We have received a lot of questions. So I'm just going to, okay. So we have received a lot of questions, and a lot of them have come from this particular part of your presentation when you were talking about the solvent models and the water model. So okay. I'm just gonna put all those questions together and ask you. So the first question is, like they want you to elaborate a little bit about the solvent models, and also like what would be like, how do you choose like different solvent models, the water models? Why did you use tip 3P water? Why did you show that? And is there somewhere they can read up about it, about the different solvent models in Schrodinger?
Yes, sure. So let's break this down. So there are different water models, and the reason why there are different water models is because of 2 things. One is how long does it take to compute the interactions, and 2 is when was the water model developed. So if you look at the different water models, some of them are 3-point water models, some of them are 4-point water models, some of them are 5-point water models. So a point indicates the number of atoms that you might have in your system. So for a 3-point water model, we have the oxygen, hydrogen, and hydrogen, which is what you would expect for a water model. But if you also think of a water model, the oxygen has two lone pairs. And there are different ways of trying to account for the lone pairs. And that's why you have different water models, like 4-point water models or 5-point water models. The more the number of points that you have, the more the number of calculations that you have to do. So 3-point water model will have like probably 2 or 3 calculations. A 4-point water model will have an extra calculation that you have to do. And it's not just for one water molecule, you have to do the calculation, extra calculation for all the water molecules in your system. And because you have to do the calculation for all the water molecules, the extra calculation, it's going to be computationally more expensive. So again, it always comes down to how much computing power you have and what is the speed that you get with each water model. A 3-point water model like SPC or T3P is the fastest. And a 4-point water model is going to be slower than a 3-point water model. A 5-point water model is going to be slower than a 4-point water model. So you have to take these things into account when you're trying to do a simulation. In most cases, a 3-point water model or a 4-point water model are sufficient and they are accurate. SPC was one of the first water models that you had, and T3P came after SPC, so it has better properties compared to SPC. TIP4P models are even newer compared to TIP3P models, and so they are more accurate. But you also again have to think that the 4-point water model is going to have an extra calculation, and it's going to be computationally expensive. There are new water models like OPC, and so there are different things that you have. So it always is a decision that you have to take depending on which water model is accurate. TIP3P is the best one, or SPCE, not SPC. SPC with an extended water model is the best one that you can use. And so there are different choices that you have to make. And if you want to get more details on water models, you can look at Wikipedia. Wikipedia has a lot of information. You can just Google all the water models and you have all of this information that are there. And TIP3P or TIP4P EW are good water models that you can use for these calculations.
Right. There's another question. So is there a particular water model that is compatible with OPLS3E?
Yes, TIP3P or SPC are compatible with them. You'll have to look at the research papers. I can get back to you in detail as to which one is the best compatible one, but TIP3P and TIP4P should work perfectly well with OPLS3E.
All the water models are very well parameterized in OPLS3.
Thanks, Pritesh.
Right. So I think we covered all those questions. Since there were so many repetitions, so I thought maybe we can cover them all together. So the next question is, why do you take 1 to 5 femtoseconds time step usually? Why is it preferred?
So the reason why you have to take a small time step, like you can probably try 10 femtosecond, but it's not recommended and it's not ideal either. The reason being that if you look at the CH bond frequency, the carbon-hydrogen bond frequency, it has a particular frequency for that, like tempo -14 or -15. And so if you have a time step bigger than 1 to 2 femtoseconds, or even 5 femtosecond, then what would happen is if you try to move your atoms, by the time you move your hydrogen and carbon atoms, the hydrogen and carbon bond will be broken if you take a bigger time step. And so you have to restrict it to 1 to 2, 4 or 5 femtosecond because you want to keep the hydrogen attached to the carbon, and the CH bond frequency is of the order of a few 10 femtoseconds or 20 femtoseconds. So you want to have your time step below that value.
Thank you. So the next question is, I think you mentioned something like you can run up to 100 nanoseconds a day. So on what hardware are you giving those figures?
These are on regular gaming cards like GPUs. Like these numbers are applicable for any of the GTX series. The P100s or the V100s have even faster time numbers. So these are just standard GPU cards that you can get that you use to run your computer games or any of those things. And these numbers are available. You can look at the Schrodinger website to find out the timings for each of the different GPUs that are there. Just look at the website and you'll get to know like what GPU will give you how much speed and what is the number of atoms that you have in your system. It is all parameterized. It's all benchmarked. You can just look at the table that is available on the website.
Thank you. So the table is available at Schrodinger.com/Desmond. So you can just go to that page and there is a nice graph there that you can look at. So the next question is about the hardware requirements. I think we got a few questions. So the hardware, you require a Linux and a GPU card that is supported by Schrodinger. If you go to schrodinger.com/supported platforms, I will also put these link in my announcements later, so you can just look at that to get more info on the hardware part. Dr. Kaushik, there is a comment sort of a thing. So the person says, I think we need to work in reverse. First, we need to do the molecular dynamics followed by IFD, then the molecular docking. Because when we go for MD, there is a lot of crucial ligands that basically we take them out after the MD. So what do you think? Is that the right way? Is that a way?
It depends on the number of ligands that you have. If you have thousands of ligands, then you should never do MD simulations for thousands of ligands because you're just wasting a lot of computing power. In those cases, you always have to do docking first to narrow down. And docking is good enough. It's not perfect, it's good enough. And you have to reduce it to 20 or 30 compounds. And when you have that, you can go for MD simulations. If you directly start with MD simulations before doing docking, if you have 10 ligands, yes, maybe you can do it. But if you already do MD simulations, then you don't have to do docking after that. MD simulation is good enough for you to understand if your ligand is interacting with the protein or not. And you can also do MMGBSA calculations to identify an accurate binding energy for your molecule. So if you're doing MD simulations first, I don't think you should do docking after that. Docking should always be before MD simulations. I wouldn't really do it in reverse order. I would always do it from docking and then you go for MD. You don't have to do IFD either. You can go from docking to MD directly if you have the computing power and if the number of ligands are 10 or 20, not more.
Thank you. So the next question is, why is it necessary for MD systems to be neutral?
That's a good question. And the answer is not simple. The reason that you have to keep the system neutral is because of periodic boundary conditions. And so think of this, when you do an experiment, the protein is mostly in a neutral environment. And they make it neutral by adding salt, so that it tries to keep it neutral. But if you have an ion in MD simulation, then that ion is going to be replicated in the periodic images. And because you have the ion being replicated in periodic images, then, so let's say you have one ion or two ions, then it's a +2 or -2 charge. Then that -2 charge is replicated in all the images. And so your overall system will have a really high positive charge, a really high negative charge. There are ways to overcome this. So the most methods right now use particle mesh evolved to account for the effects of long-range interactions because of the charge. And if you have a big box like a buffer of 12 angstrom or 13 or 14 angstrom, 15 angstrom from your protein or ligand atoms, then PME accounts for most of those net charge. And so it's not necessary for you to have a neutral molecule, but it's recommended to have a neutral molecule if you are going to have a smaller box and if you're not using PME. But I think most cases like these days, you can get away with it, but it's recommended to have a neutral system. And the best way to neutral a system is by adding salt. If you're adding ions, then you should also add salt. Otherwise, you have a net charge that is duplicated across all your periodic boundary images.
Thank you, Dr. Kaushik. I think you answered the next question also. So the question is, when do we need to add salt and when we can avoid this?
Just to add on that point, you always want to replicate your MD simulation to be as close to the experiment. And if when you look at the paper that they did experimental studies in them, and if they use salt when they did the calculation, then you have to use salt in your simulation as well. So it's not just about neutralizing your system, it's also about trying to be as close to the experimental as possible. So always read the paper, always try to see what has been done before. And if there is no information on that, then you might you don't have to add a salt, but if the experiment uses salt, then you have to use a salt.
Thank you. So the next question is, what is the optimal simulation time, recording interval, and the number of frames?
So the number of frames, so the simulation time interval, ideally you want to, depends on what you want to study. So if you want to study just the stability of the ligand, like 15 nanoseconds is sufficient to do the calculation. But if you want to study ligand binding and unbinding, where it goes into the binding pocket, goes out of the binding pocket, then you have to do a calculation for microseconds. And again, the number of frames that you want to have is also dependent on what you're trying to study. For example, if you're looking at stability of the ligand, then you have to save your frames often, like every 10 picoseconds or 20 picoseconds. But if you're trying to do a calculation for ligand unbinding and binding, then the motions happen on a longer time scale, like microseconds. So you can save your simulations every 100 picoseconds or 200 picoseconds, but it's recommended to save your frames at least every 100 picoseconds or even 20 picoseconds, because if you have more number of frames, you can always skip the frames when you're doing analysis. If you have less number of frames, you can never add frames to it. So you always want to have as many frames as possible, but a good number is 10 picoseconds or 20 picoseconds is a good number to save your frames. And the simulation timeline is always dependent on what you're trying to study.
Thank you. So moving on to the next question, how do we choose between different ensemble classes?
So the ensemble classes are, so I mentioned you 5 different ensembles, but you mostly will be dealing with NVT or NPT. If you have membrane, then you have NPAT or NP gamma T. So you only use those ones if you have a membrane, the last 2 ones. But NVT and NPT, you can use either one of them. NPT is always recommended because NPT is close to what the experiment does. When people do experiments, they do the calculation at atmospheric pressure and at 300 Kelvin. You can change your pressure, you can change your temperature, but it's recommended to use the default values. And because the experiment always does it at 1 bar and 300 Kelvin, you want to be close to experiment and you want to use NPT. But you can use NVT or NPT. It's not going to be a big difference. Yeah, you can use either one of them. But NPT is better because you can directly get Gibbs free energy from an NPT ensemble.
Right. So the next question is it possible? Okay, is it possible to run MD on a simple ligand molecule to see the interaction between the surrounding water? And is it possible to change the solvent as well?
Yes, you can do just a simple ligand in a water box. You can also do a simple ligand in different solvents too, like urea or octanol or methanol. You can use any other solvent that you want to use. You can do all of those things, and it's possible with Schrodinger and also other softwares too.
Right. The next question is, how much RMSD deviation is permitted? Do we need to consider the mean RMSD or the individual point RMSD?
So your RMSD should ideally be less than 2 angstrom from your initial structure or from the native structure or extra structure. You can probably go to maybe 2.5 to 3 for the protein, but the ligand should be less than 2 angstrom. Again, these are not like strong cutoffs. It's recommended to have them less than 2 angstrom. You need to look at the individual data points for the RMSD. Looking at the mean RMSD does not tell you that much, because let's say you have a ligand that just unbinds and binds again. And so if it's binding and unbinding, then you can have an RMSD of 5 angstrom. And if it binds, you can have an RMSD of 2 angstrom. And so the average would be around 3.5, something like that. If you're trying to publish your data, it's always recommended to have the individual RMSD points for your entire simulation in either the main paper or in the supporting information.
Thank you. There's a question, is MD simulation done on Galaxy, that is use Galaxy platform? Does that work better?
I'm not sure of what the US Galaxy platform is. Do you have any idea?
I'm not sure, but I think it's a different platform other than Desmond to do MD.
No,
I'm not sure, but I think we can only answer about Desmond here. Yeah, right. Okay. The next question is whether we have to perform docking before MD. Are we using the output of docking study for MD simulation studies? And also,
I think I went through that before.
I think, yes, that is what we were primarily discussing. But you can also do ligands, as mentioned before. So the next question is, can we do protein-protein MD simulations on Desmond?
Yes, you can do protein-protein MD simulations. You can do protein RNA. You can do protein ligand. You can do protein DNA as well. You can do for simulations for any molecule that you can think of.
Okay, another question is again on hardware a little bit. So it's just a general question. Can we run Desmond in virtual partition having Linux installed in a Windows system? The answer is yes, but you need to have the right GPUs. The other question is, why do we need to modify the protein? Because we modified during in silico. Later, in the case of in vivo, how the ligand was stable. Do you understand the question? Should I repeat it again?
No, I'm not sure what the modification for the protein is in this particular case is.
Yeah, I think it could be just a general question. Maybe came
we
from...
always need to prepare the protein before you do the calculation. But if you are talking about why we need to do the simulations, then the protein is going to move, and it's also going to move in vivo as well. It's never a static structure in vivo also. So if the modification is pertaining to the protein structure, then simulations will help. But I don't understand the question perfectly. So
yeah. If you want to, you can just email us the question if we did not understand it properly. Okay, sorry, these are just the new questions. I have not read them before. Okay, so proteins in human physiology stay in a variable ionic environment. Then how we keep a neutral environment?
Yes, so in simulations, you have the